Hey everyone, welcome back to the channel. Today we're diving into something wild that's been popping up in the news. A new bat coronavirus called HKU5-CoV-2. Yep, you heard that right. Another coronavirus, and it's got people asking, is this the next COVID-19? Are we heading for round 2 of pandemics? Well, don't freak out just yet. I'm here to break it all down for you, step by step, with the latest info we've got. If you're new here, this is where we unpack the science behind the headlines in a way that's easy to get and honestly pretty fun. So hit that subscribe button and let's jump into the world of HKU5-CoV-2 together. Okay, let's start with the basics. What is HKU5-CoV-2? Picture this. Scientists in Hong Kong are out there studying bats, specifically the Japanese pipistrelle bat, a tiny little guy you'd barely notice flitting around at night. And in these bats, they find a new virus. That's HKU5-CoV-2. It's a coronavirus, which means it's part of the same family as the viruses behind SARS, MERS, and, yep, COVID-19, SARS-CoV-2. But don't let that scare you off the bat, pun intended. Not every coronavirus is out to ruin our lives. So, why is it called HKU5-CoV-2? The HKU part stands for the University of Hong Kong, where some of this research kicked off. The 5 ties it to a specific lineage of viruses they've been tracking, and CoV-2 just means it's a coronavirus, version 2 in this naming scheme. Not directly related to SARS-CoV-2, though they share some traits. It's classified as a merbecovirus, which is a fancy way of saying it's in the same subgenus as the MERS virus. MERS, if you don't know, hit the Middle East hard back in 2012 with a scary 35% fatality rate, though it didn't spread as easily as COVID-19. Here's where it gets interesting. HKU5-CoV-2 uses the same doorway into human cells as SARS-CoV-2. That doorway? It's called the ACE2 receptor. Think of ACE2 as a lock on the outside of our cells. Lungs, heart, intestines, you name it. The virus has a spike protein which is like its key. If that key fits the lock perfectly, the virus can slip inside and start multiplying. SARS-CoV-2 was a master locksmith. It fit ACE2 like a glove, which is why it spread like wildfire. HKU5-CoV-2 has a key too, but it's more like a bent, rusty one. It can unlock the door, but it's not nearly as good at it. Scientists call this weaker binding affinity, and it's a big reason why they're not sounding the alarm bells just yet. But let's not gloss over this. Bats are basically virus hotels. They host over 3,000 types of coronaviruses, and most of them don't bother us. Why? Because jumping from bats to humans isn't easy. It often needs a middleman, like another animal or a mutation that makes the virus better at infecting us. So, while HKU5-CoV-2 has potential, it's not primed for prime time, yet. Now, how did we even find out about HKU5-CoV-2? This is where the story gets cool, and a little gross. The discovery comes from a team led by Xu Zhengli, a superstar virologist from the Wuhan Institute of Virology. If you've heard of her, it's probably because she's nicknamed Batwoman for her decades of work chasing down bat viruses. She's like the Indiana Jones of virology, only instead of ancient artifacts, she's hunting microscopic pathogens. Her team went out into the field, think caves, forests, anywhere bats hang out, and collected samples. Specifically, they took anal swabs from these Japanese pipistrelle bats in Hong Kong. Yep, bat poop samples. It's not glamorous, but it's how you get the goods when you're studying what viruses these critters are carrying. Back in the lab, they used a technique called PCR, polymerase chain reaction. If you've ever done a COVID test, that's the same tech. It's like a magnifying glass for DNA or RNA, letting them spot the virus's genetic code in all that bat gunk. Once they confirmed they had something new, they didn't stop there. They grew the virus in the lab using cell cultures. Think petri dishes full of human cells tweaked to mimic our lungs or intestines. These cells were engineered to have ACE2 receptors, so they could test if HKU5-CoV-2 could break in. Spoiler alert, it could. They also used organoids, which are like mini organs grown from human cells, tiny lungs and guts in a dish. Again, the virus could infect them. But here's the kicker. This is all in a lab. Just because it can infect human cells under perfect conditions doesn't mean it's ready to jump into the wild and start a pandemic. It's like saying a car can drive in a showroom. It still needs gas, a road, and a driver to go anywhere real. Let's geek out a bit and dig into the science. Why does HKU5-CoV-2 have people talking? It's all about that spike protein and how it interacts with ACE2. The researchers used a high-tech tool called cryo-electron microscopy, cryo-EM for short, to zoom in on this interaction. Cryo-EM freezes the virus and receptor together, then takes super detailed 3D pictures. It's like getting a snapshot of a key sliding into a lock at the molecular level. What they saw was that HKU5-CoV-2's spike protein binds to ACE2 in a way that's similar to SARS-CoV-2 but not identical. There are subtle differences in the shape and chemistry that make it less snug. Imagine trying to plug a cheap knockoff charger into your phone. It fits, but it's wobbly and doesn't charge as fast. That's HKU5-CoV-2. It can get in, but it's not optimized for human cells like SARS-CoV-2 was. Another big deal is something called the furin cleavage site. This is a little spot on the spike protein that acts like a secret weapon. Furin is an enzyme in our cells that snips the spike at this site, making it easier for the virus to fuse with the cell membrane and sneak inside. SARS-CoV-2 had this, and it's one reason it was so infectious. HKU5-CoV-2 has a furin site too, which raises eyebrows because it could mean the virus has room to evolve into something sneakier, but right now, that weaker binding to ACE2 is holding it back. Here's where it gets dicey. Viruses mutate. All the time. Most mutations are duds. They don't change much, but every once in a while, a mutation hits the jackpot and makes a virus better at spreading or infecting. SARS-CoV-2 gave us variants like Delta and Omicron because of this. Could HKU5-CoV-2 pull the same trick? Maybe. That's why scientists are keeping tabs on it. Okay, let's tackle the big question. Should we be worried? The short answer, according to the experts, is no, not right now. Xia Zhengli and her team have been clear. The risk of HKU5-CoV-2 causing a widespread outbreak is low compared to SARS-CoV-2. Let's break down why. First, that weaker binding affinity we talked about. It's a huge hurdle. A virus needs to stick to our cells efficiently to infect us and then spread from person to person. HKU5-CoV-2 isn't there yet. Second, there's no evidence it's jumped to humans outside the lab. 0 cases, 0 outbreaks. Third, even if it did, it might not transmit well. SARS-CoV-2 was a champ at going airborne. HKU5-CoV-2 might not have that knack. But, and this is a big but, we can't just ignore it. Coronaviruses are sneaky. They've got a knack for evolving, and history shows they can surprise us. SARS in 2003 came from bats via civets. MERS jumped through camels. COVID-19 might have involved pangolins or something else. We're still piecing that puzzle together. The point is, HKU5-CoV-2 could theoretically mutate or find an intermediate host that makes it better at infecting humans. Then there's the zoonotic angle, how viruses spill over from animals to us. Bats don't usually hang out with humans, but if someone's hunting them, handling their droppings, or living near a bat colony, that's a chance for contact. Add in things like deforestation or wildlife trade, and you've got more opportunities for a virus to say, hey, humans look like a nice new home, so the risk is low, but it's not zero. That's why surveillance is key. Watching bats, testing samples, and keeping an eye on any weird respiratory outbreaks. It's like having a smoke detector. You hope it never goes off, but you're glad it's there. So, what does this mean for us as a society? How do we handle something like HKU5-CoV-2? Step 1. Better surveillance. Scientists need funding and tools to keep tracking viruses in the wild. That means more bat swabbing missions, more labs sequencing genomes, and more data shared globally. The earlier we spot a potential threat, the better we can prep. Step 2. Education. People need to know the risks of messing with wildlife. In some places, bats are food or medicine. Cultural practices we can't just dismiss. But we can promote safer ways to handle them, like wearing gloves or avoiding contact altogether. Same goes for guano, bat poop's used as fertilizer, but it's a viral goldmine. Simple stuff like masks or better sanitation can lower the odds of a spillover. Step 3. Global teamwork. Viruses don't care about borders. The World Health Organization, WHO, is like the planet's virus watchdog, but it needs countries to pitch in, share info, fund research, build stockpiles of supplies. COVID-19 showed us what happens when we're caught off guard. Shortages, confusion, delays. We can do better, and let's talk ecosystems. Bats aren't the bad guys here, they're just doing their thing. But when we chop down forests or crowd into their habitats, we're rolling the dice. Conservation isn't just about saving cute animals, it's about keeping viruses where they belong, in the wild, not in us. Now let's say HKU5-CoV-2 does start causing trouble. What's our game plan? Good news, it's a coronavirus, so we've got some playbook pages from COVID-19. Antiviral drugs like remdesivir might work. It jams up the virus's replication machinery, and since coronaviruses share some of that machinery, it's a decent shot. Same with monoclonal antibodies. Those lab-made proteins that latch onto SARS-CoV-2's spike might cross-react with HKU5-CoV-2 because of the ACE2 connection. More tests would tell us for sure. Vaccines? We're in a golden age for that tech. mRNA vaccines, like Pfizer and Moderna's, could be tweaked to target HKU5-CoV-2 if needed. Same with adenovirus vaccines like AstraZeneca's. The trick is the spike protein. If we can train our immune system to recognize it, we're golden. But here's the reality. Vaccine development takes time and money, and with the risk low right now, it's not top of the list. Still, having that capability ready to roll is a huge win post-COVID. Prevention's the real MVP, though. If we stop the virus from jumping to humans, we don't need to scramble for cures. That's where surveillance and education come back in. Cheaper and easier than a global vaccine campaign. I get it. This kind of news can hit hard. After COVID-19, anything with coronavirus in the name feels like a gut punch. Lockdowns, masks, losing people we love. It's fresh and it's scary. So when HKU5-CoV-2 pops up, it's natural to think, here we go again. But let's breathe and look at the facts. The scientists are on it. They've been crystal clear that this isn't another SARS-CoV-2, at least not yet. No human cases, weaker binding, low risk. That's not downplaying it. That's just where we stand. Fear can spiral fast. Remember the toilet paper hoarding? But it doesn't help us. What does? Staying informed. Check reputable sources like the WHO, CDC, or peer-reviewed studies, not random X posts or clickbait headlines. That said, preparedness matters. COVID taught us we need strong healthcare systems, quick testing, and clear communication. HKU5-CoV-2 is a reminder to keep those lessons alive. And you can do your part. Wash your hands. Seriously, it's a superpower. Skip the bat stew and keep your vaccines up to date. Small moves, big impact. All right, let's tie this all together. HKU5-CoV-2 is a new bat coronavirus with some tricks up its sleeve, like that ACE2 key and furin site. But it's not primed to take over the world. Its binding's weak, there's no human spread, and the risk is low. That's the latest scoop as of December 2024, and science moves fast, so we'll keep watching. Key takeaways? Surveillance and research are our best friends. Education and global teamwork can stop spills before they start. And if push comes to shove, we've got tools from the COVID playbook to fight back. It's not about panicking, it's about staying sharp. Science isn't static. New studies might tweak this story, so keep your eyes peeled. For now, HKU5-CoV-2 is a heads up, not a red alert. Stay curious, folks. It's how we stay ahead. That's a wrap, everyone. Thanks for sticking with me through this deep dive into HKU5-CoV-2. If you liked it, smash that like button, share it with your crew, and subscribe for more science breakdowns. I've got plenty more where this came from. Got questions? Thoughts? Drop them in the comments. I read everything. Stay safe, stay curious, and I'll catch you in the next one.